Hello students, welcome in the world of biology. In this video lecture, we will be discussing about the molecular scissors. Molecular scissors means restriction enzymes. They are very essential tool in rDNA technology. And we know that all the higher organisms, eukaryote, the plants or animals, has some or other form of defense mechanism. But when I think of bacteria, I feel very pity on them, knowing that do they have any defense mechanism? Bacteria can be infected by viruses, which are called as bacteriophages. Bacteriophages are the viruses that infect the bacteria. But does this bacteria have any protection against them? So yes, restriction in the nucleases are the part of bacteria's defense against invaders. What happens when bacterial phage infects a bacteria, injects the DNA into the bacteria? If that viral DNA replicates, it will assemble into new virus, thereby killing the bacteria, either in the same generation or in the other generation. What happens is bacteria can synthesize an enzyme which cleaves DNA, which cleaves nucleic acid. That's why it is called as nucleases. Now, as these enzymes can cleave the DNA, it can also cleave the bacterial DNA because it's also exposed. Bacterial DNA, can remember, is not bounded by the nuclear membrane. So, to protect its own DNA, bacteria modifies its own DNA by attaching the methyl group. So, this system works as restriction and modification system. That's how a class of enzymes were discovered where the enzymes could cut the DNA. Now, there are two types of enzyme, restriction enzymes. One that cleave inside the DNA are called as restriction endonucleases, and other that can cleave DNA at the ends is called as restriction exonucleases. This more than 3,000 of restriction enzymes has been identified, and more than 600 are available commercially. They are used to modify the DNA, and the manipulation for DNA in laboratories. They are essential tool for the recombinant DNA technique or biotechnology. Now, these restriction enzymes act at a specific sites which are called as restriction sites. When the restriction sites are present, then and then only restriction enzymes will be able to cut the DNA. Otherwise, it will not be able to cut the DNA. And these restriction sites are mostly the palindromes. We know that palindromes are the words that read the same as forward and backward, like Hana, level, L-E-V-E-L. Whether you read it from backward, you read the same, or from forward, you read the same. Now, such type of sequences are present in the DNA. Those are called as palindromic sequences. If you read those sequences from 5' to 3' and from 3' to 5', they will read the same. And these sequences, palindromic sequences, are often recognized by the restriction endonucleases. So, those are called as restriction site. Now, remember, in the second slide itself, we have seen the restriction modification system, in which I told you that bacteria modifies its own DNA to protect its own DNA. So, they modified or they mask the restriction site of their own, modifying the nucleotides with the help of attaching the methyl group. Whereas viral DNA restriction sites are exposed and restriction enzymes can cut the viral DNA. Hope this lecture was useful to you. Feel free to ask your doubt by commenting on this video. Please subscribe it for my latest update. Thank you.